neurotoxins on the high-throughput automated electrophysiology instrument IonWorks Barracuda Plus System. So just a little bit of the background. So we all know that the neurotoxins, or the toxins from animals, plants, and microorganisms has been playing a very critical role in the understanding of ion channel function and structure. During evolution, the venomous and poisonous organisms turned out to be excellent pharmacologists of the nature, since their toxin evolved to be highly specific for targets in the CNS or PNS, central or peripheral nervous system. Examples shown here are the toxins from jellyfish, from spider or tarantula, from snake and cone snails. And depending on the types of toxins, they can be working on different ion channel types, including the potassium channel, sodium channel, and calcium channels. And so the mechanism of actions can also vary from modifying the gating machinery to plugging the hole or the pole, and modulating the inactivation property as a biophysical property of the ion channels. As a matter of fact, one of the conotoxin, one of the toxins, has been developed into FDA-approved drug for managing chronic pain. This is called ziconotide, which is a synthetic peptide made from the cone snail toxin, and it has a potency for greater than morphine for 100 to 1,000 times. Just showing an example here that there are actually 700 species known to date that just from the cone snail, this species by itself, and each contains up to 200 different venom peptides. So implying there's a huge library of more than 100,000 compounds of possible pharmaceutical interest that is potentially available in their venom. This is taken from a review article by Stevens et al. In 2011. So it's the toxin, neurotoxin as a group has great potential in becoming drug candidates for a multitude of human diseases. However, the screening or the electrophysiological characterization, or especially the high throughput electrophysiological characterization of the neurotoxins have been challenging for many reasons. A few are listed here, including the physical chemical properties of the neurotoxins. They typically are fairly large, with molecular weights in thousands of daltons, in several kD, versus small molecule compounds in hundreds. And also different neurotoxins may have different hydrophobicity. So their interaction with the ion channel and with the cell membrane can be highly diverse. This has an implication on the toxin in channel interaction, in the sense that the ion channels typically present several binding sites for different types of toxins, and depending on the different binding sites, the affinity of the toxins to the channel can be highly variable. And also, what makes it a little tricky to analyze the electrophysiological properties of this interaction is the binding of the toxin to the channel can be highly dependent on the state of the channel, or the voltage of the the membrane potential of the channel. So these voltage or state dependent binding affinities require highly sophisticated control of the membrane potential to analyze the functional impact of the toxin to channel interaction. And also, in some cases, the binding of the toxins to the channel can be fairly slow. Compared to small molecules, this can take tens of minutes to reach the peak modulation effect. So, given the challenge we have right now to characterize the neurotoxins, what we strive to present here is a case study where we use IonWorks Barracuda system, which is a high throughput automated patch clamp system to analyze this neurotoxin pharmacology. Right? Just a sort of a very brief introduction or reintroduction of the system. The Barracuda has 384 recording sites, parallel recording sites, and it has 384 discrete patch clamp amplifiers and fluidic channel pipettators. It can be run in both single hole, which records a one single cell at a time, or a PPC, which records ensemble signal of 64 cells per recording site. And also, what makes the Barracuda very attractive is this highly sophisticated fluidic design, which allows the precision and accuracy of compound addition to the cells, to the ion channels. So without further ado, I'll show some of the examples where the Barracuda can be utilized to tackle the pharmacology of neurotoxin. So one of the features of Barracuda is actually allows very sophisticated programming of the voltage protocols, and in a mode we call the assay development mode, the customer can actually run several different voltage protocols using the same batch of cells and the same patch plate in a way very similar to the manual patch clamp rig, and optimize their voltage protocols to the best of their expectation. Showing here is an example where we use the same batch of cells and apply different voltage protocols to characterize the voltage dependent activation, voltage dependence of inactivation, and also the recovery from the inactivation for the sodium 1.5 channels, which is the target of choice for this study. And it's showing the characterization of the biophysical parameters for both activation and inactivation. And once you have the knowledge of the channel, you can program protocol that best suits your need. In this case, we have decided to use 3-second 10 Hertz trend shown here on the bottom. So a total of 30 depolarizing pulses in the trend. And the current recorded from such a voltage protocol is shown here, where the sodium current shown in the if you zoom in, you can see the very nice resolved kinetics of the sodium channel activation and inactivation from both the pulse 1 and pulse 30 of the trend, and the amplitude of the current are fairly stable within the trend. So in addition to the sophistication of the voltage protocol, for any pharmacological assay, especially for compounds with slow on rate or off rate, it's highly desirable to have a stable assay window. And what we're showing here is the proof principle study where we showed the current, in this case the sodium 1.5 current, remains stable for up to 30 minutes since the beginning of the recording. So what's shown here is, the right bar here each indicates one pulse in the 30 minutes in the 30 pulse trend. So the arrow here indicates the addition of the just buffer solution, external buffer solution at the different time point, and it shows like over 30 minutes the current remains fairly stable, not only within the same trend, but also at different time points. So having a stable assay current for up to 30 minutes really allows us to tackle the compound with slow kinetics. And one of the examples that was shown previously in the last webinar that Jim presented is the utility of this long assay window to measure both use-dependent inhibition by several classic compounds like lidocaine and tetracaine, and also the off rate of the compound by applying fresh buffer to remove the binding of the compound from the channel. So just to briefly recap, this is a control group where the current remains stable for up to 30 minutes, whereas in the group of cells that's treated with tetracycline, you can see immediately after the application of the tetracycline, there's a very striking use dependence where the first pass of the train, the current remains fairly large, and toward the end of the train, the 30th pass, there's almost nothing left. And upon the application of the fresh buffer to remove the binding of the tetracycline, you can see a gradual recovery of the peak current to almost the baseline level. And this is more apparent for the another compound, lidocaine, the use dependence block, and the removal of the use dependent block, and the recovery of the current to the baseline level. This is showing TTX, not so much of use dependence, but can be the current can recover to the baseline level fairly nicely. Okay, so what we are trying to show here today is another example we think can be potentially applicable in the drug discovery field, is to utilize a long-acting window to analyze the voltage and the state-dependent modulation of the neurotoxins, and also the slow on-rate of the neurotoxins to the sodium channel. So a brief introduction to the experiment design. So here we still use the human sodium 1.5 channels expressed in the CHL cell line, and we chose 3 different toxins, 2 from tarantula toxin. One is called a gingival toxin 2, which has a pronounced effect in slowing down the inactivation of the sodium channel, and is selective to sodium 1.5 channel. The other one is called protoxin 2, which is a blocker of the sodium 1.5 channel. And as a negative control, we chose toxin from muconotoxin P3A, which is selective for sodium 1.4 channel, and have, according to literature, very little effect on the 1.5 channels. So we made all this compound in the final solution containing about 0.3% of BSA. And we're still using this 10 Hz 30 pulse train to analyze the modulation of the channel by the neurotoxins. So just kind of give you a visual understanding of the experiment design. We start with 3 baseline scans, with each scan is 3 minutes apart from the neighboring one, and then apply the compound, in this case the number of toxins, and a DMSO control as well. And then we perform 10 post-compound scans, again with 3 minutes in between each scan. So the whole assay is about 35 minutes long. Okay, so let's look at some of the data. So this is showing you the recording of the sodium channels, but only showing the first pause in the trend. So so you can have a good appreciation of the channel kinetics. So first of all, there's almost 100% success rate, except these 2 wells, which we know coming from hardware. We have hardware errors in those 2 wells. But the rest of the plate all give you very nice current, and also because we're running this in the PPC mode, you're seeing that the uniformity of the current amplitude is very good. Moving into the post-compound scan. This is post-compound scan 2, which reference about 4 minutes into the compound addition. And you see in the group of cells that's applied with gingival toxin, you see a very striking difference between the control and the drug-treated ion channel currents. One is the most outstanding observation is the slowdown of the inactivation, and there's some moderate enhancement of the peak current as well. In the group that's challenged by the protoxin 2, you see essentially the majority of the current is inhibited. And whereas compared to the negative control, the immunotoxin or the vehicle control DMSO, there's virtually no change of current amplitude. So this is about 4 minutes into the compound addition, and the second represents about 15 minutes into the compound addition. This is post-compound 6. And again, you're showing the neurotoxin behaves very different than protoxin or the control group. So to have a better view of the effect of neurotoxin, I recreate the current profiles before the compound addition and after the compound addition. So what's shown here is in black is the control current, in red is the post-compound scan 2 or 4 minutes after compound addition, and in the purple is the 15 minutes after the compound addition. So you're saying two things. One is there's a moderate increase of the peak current, and then there's a significant slowdown of the inactivation of the sodium channel. So to compare the data with the manual patch computer recording, this was data supplied from the alimony labs, where the compound or the toxins were purchased from. This is the time course of the neurotoxin action on the sodium 1.5 channels. So it virtually takes about 7 minutes to reach the peak effect of the modulation. And for us to analyze the effect of the neurotoxin, we picked the two metrics. One is the peak amplitude of the current, as shown here. One is the amplitude of the persistent current, which is shown here at this time point. So on the control conditions, about 7 to 8 milliseconds after the channel opens, the persistent current goes back to almost zero, but certainly not in the case where the neurotoxin is applied. Okay, so again, showing what a control group, how the control group the current remains stable for up to 30 minutes. This is the pre-compound scan number 3, where this is showing the time point, and after the compound addition, the current remains fairly stable for up to 42 minutes, so 35 minutes after the beginning of the data acquisition. The difference, there's about 12% of current rundown compared from the very beginning of the experiment toward the very end. So it's reasonably good. So what does neurotoxin do then? So compared to the negative control in conotoxin and the vehicle control in DMSO, neurotoxin exhibited a relatively bigger enhancement of the peak current. So this is looking at the peak amplitude of the current, whereas immediately after the compound addition, there's really no difference. And 10 minutes afterwards, you're seeing the peak effect of the enhancement of current amplitude. But if you look at the persistent component of the sodium current, the modulation by the neurotoxin is very pronounced. As I mentioned earlier, at the time point we used to measure the persistent current, typically there's very little, about 2% of the total current left, 7 milliseconds after the channel open. And in the case of neurotoxin, where you see there's two things. Number one, a very pronounced enhancement of the persistent current. And number two, there's a time dependence where the enhancement of the current reaches the peak amplitude at about 10 minutes. 10 minutes after the compound addition, which is very in alignment with the very much in alignment with the manual patch clamp data that was shown before. So what about the protoxin? In this case, again, we're looking at the current stability in the vehicle control and in the conotoxin negative control. Looking at compared to this, looking at the protoxin, you say immediately after the compound addition, there's sizable reduction of the current, but the current didn't, reach the maximum. The current wasn't maximally inhibited until 10 or even 20 minutes after the application of the compound. And something is really interesting is that showing here is. Remember we're looking at the peak amplitude of the current from all 30 pulses within that TRIM protocol, and you're saying at the beginning the very first pulse, or pulse 1, has the highest amplitude of current, whereas the last one typically have a lower amplitude of the current. So 10 minutes after the protoxin application, you're seeing a quite opposite effect, where the protoxin is the P1 has the smallest current, whereas the P30 has the largest current. And this is not an artifact, as it has been repeated 3 times in the following measurements at different time points. So this would suggest that the binding of the toxin is somehow removed from the channel during the TRIM protocol. And in looking at the published literature, there's actually evidence just exactly supporting the hypothesis. So this is an article taken from Sokolov et al. Published in 2007, where they analyzed the binding of the protoxin to the sodium channels 1.2 and 1.5, and they concluded that, number 1, the toxin binds to the voltage sensor of the domain 2 of the sodium channel, and strong depolarization actually reverses the binding by removing the protoxin from the channel binding site. So a few examples shown here. They're not using exactly the same protocol we're using, but they're showing both the time dependence and the voltage dependence of this reversal of protoxin from the sodium channels. So this is very interesting to us. I mean, we started the experiment without knowing that the protoxin would behave differently than typical sodium channel blocker, and having the ability to capture the reversal of the channel inhibition provides insight into the mechanism of action, which is supported by the literature already published. So just a quick summary, the IonWorks Barracuda system has very sophisticated electronic and fluidic designs, which supports both sophisticated assay development and the long assay window for analyzing of parameters like on and off rate of different compounds, as well as the voltage and state dependence of the binding of compounds to the channel. So that's all I have, and I'll be happy to take any questions toward the end of this webinar. Thank you.